Actually,
So what is
It's still logging. I get those too. It's like right. I said as well. I wasn't really hard until I was tired. Yeah.
It was?
It was
I was in the library all night.
Oh, wow.
Yeah.
Yes, I'm a great one.
I turned into.
I like, oh my god. It's like that little bear they had when we were. Okay,
He's like, oh,
but it's in case the kids get started or just.
That's actually really exciting to me. I just feel like the universe is telling you to
I don't even know about it unless I was in
but I like that's really funny actually.
as a consultant. It's just, it's new. It's also an Asian
It's like girls.
We don't know that.
Like this close to it. Oh. Now I'm a little bit less close to it. I wasn't. I but it's not and now my doctors are like, oh, I do too. I want to make sure though, like, that'd be so bad.
That's right. I think that's right.
What do they say?
Today, maybe one of the last days we'll be talking heavily about chemistry. Today we're going to what we really want to show off is that it's a really powerful tool. It has a very binding of things, and depending on what it binds to, which you're going to see throughout today, is going to do that. It changes its ability, right? So we're going to be able to change what it interacts with, or how it behaves, and we're going to get to the cool stuff where it's like you can use it to hold information like our DNA. Let's go. All right, so let's go first, right? Carbon is super important. We're gonna find it everywhere. In this figures, carbon is your, in this case, it's the red one in the center. You can see it binds basically four little structures. These are your hydrogens. Here we're talking about a carbohydrate. This is basically a sugar. So you see, now it's the black ones. It's basically our sugar chain going there. In protein model, here they're showing it in gray. You can see proteins are huge. They're long chains of amino acids. But what we want to see is just the critical amount of carbon that's found in the structure. Okay? And then finally, the one on the right, super critical one, this is the one that holds your information. It's the one that tells you basically how to make every single organ in the body, how to make a cell, proteins are making. It's your DNA. It's a little thing of the structure that kind of repeats itself over and over. And you're going to see that this gray carbon basically kind of throughout all the little structures. Here, when we actually look at the base pairs, right, A, T, C, G, you can see those we're going to find either pyrimidines, which have one ring, or purines, which have two rings. And we'll see carbon is in the center of all of those, but in different spots. Here, kind of like the color where nitrogen is showing us, hey, we have different clusters of those distributed within this part. On the back part, which is where we have the sugar and the phosphate backbone, you'll see we have a difference, which is those red and those but with that, right, carbon's just ability to grab onto things, we're able to make all these different things happen. From our information, proteins that do the work, sugars to get us energy, and methane, which is a gas we can burn off. I think you guys probably all know about methanol and ethanol, right? So the next one's going to be, how's it doing this? What's the big power behind it? For this one, remember it's on the second shell. For its second shell, it has 4 valence electrons. So that means it has 4 on its own. In order for it to achieve its valence or for it to be stable, it will need 4 more. So it has the ability to bind to 4 different molecules. And that gives us 4 places to interact with this carbon. We can have that interaction happen in a few different ways, right? You can have a molecule that has 4 single bonds, and then one electron pair being shared there, one electron pair there, one electron pair, one electron pair. This could be here. I'm showing hydrogen, oxygen. And you can also set up things where you do multiple bonds. So in this case, remember that oxygen has 6, it needs 2 more electrons to be happy. It can decide to share 2 of its own with carbon, and carbon can share 2 of its with oxygen. In that case, because we've used up the double bond, carbon has used up 1, 2, and has only 2 spots left to interact with. So you'll see 2 single bonds and a double bond, or 4 single bonds in order to make this thing be happy. So we can do single, or we can do double, even triple. Carbon doesn't do triple that much, but there's other ones that do. All right, so here we're starting with ethane, which is just carbons and hydrogens. So for this one, I kind of want you to remember when, from Wednesday's assignment, right, was whenever you guys see this, I want it in your head to be almost like you always remember CNHs, they're pretty similar electronegativity. So their interaction is going to be non-polar, right? Which means there's no charge, means this thing does not want to play around with water. Anytime you see this, you're thinking water doesn't want to get in near, doesn't want to work with this. Unless you kind of bring this over, now you've added a functional group, right? So an O and an H, now that O is going to be, when it shows up, now it has a large electronegativity, so it's going to mess with this molecule to have an area where it'll have a negative charge, and it'll give these guys a little bit of a positive charge. At that point, we have a polar bond will stick to water, and now you change the way that this works, right? All right, here's another example. You don't have to memorize it. The first time I took this class, I was trying to draw these out. But the only really important thing we're looking at is these are two sex hormones. They work in kind of opposite ways of signaling. But the only real difference are just the attachment of these functional groups. And just those differences alone are able to make them work differently. Because normally what happens is our body makes these, and you basically have receptors that will catch onto these. And through that, you can send signals to your cells that the hormones present. So literally just because you have a methyl group which is hydrophobic, that one might be able to fit into the receptor, and because that one doesn't have it, it won't fit in the pocket, so it won't sense it. So it's like you can have a really precise thing where one receptor will work with this grab on property. This receptor will not work with that one, use a different receptor that comes on with it. But it's just those functional groups giving us like this 3D shape of can we grab it or can we not grab it? All right, for functional groups, these are the ones we're kind of going to go over. The list, what I'm going to show it to you, we're going to look at it about 3 times today. The ones that I tell you how to memorize them is that everything from up here, all of this are going to be basically us bringing in those polar bonds. They're going to help us interact with water or other charged molecules. The methyl group at the end is the only one that counts into it. The hard part is I do love talking about all of these, and we're going to be talking about it today and over the next few labs. The only one I kind of don't pay that much attention to is going to be this guy. So it's going to put a little, cross it out a little bit. I probably won't pick on you guys. But the other ones, fair game, and I love talking about them. So let's dive in a little bit. All right, here we go. So this is our table. For this one, again, those 7 names that you just saw are going to be right here. We are chemical groups. And then the thing that I want you to focus on when you see this is just basically those little blue squares, right? Here we see hydroxyl group. First thing we're looking at is going to be that OH. Next is carbonyl group, which is the C, but now it's a double bond CO, right? And then when you look at the carboxyl group, that carbon, you're focused on the fact that it has a double bond with an O and an OH group. So those 3, normally I kind of remember them as a set. Yeah, so this will be the set of 3 I kind of work with, right? The top 3, hydroxyl, carbonyl, and carboxyl. The cool thing about them is they can kind of, depending where you put it on the molecule, you can change what it does, right? In this case, again, adding that ethanol group, the hydroxyl group at the end, it's going to let this thing be more soluble with water. Like you can actually mix alcohol and water, right? It's separate like oil and water. So that gives us that big power. In this case, there's a little bit extra description about these ketones and aldehydes. But the only thing they're telling you kind of is, if it's a ketone, it's in the middle of the carbon chain. If it's an aldehyde, it's at the end of the carbon chain where you pick up the double. I want you guys to remember this. I won't pick on you on these. Right? So take it easy on this and don't worry about memorizing this part. Stick to this. And then for the carboxylic group, I love using this one a lot. We use it to group proteins together. And the cool thing is, because you have two oxygens that are super negative, sometimes they'll share electrons kind of across this thing a little bit. That gives us this negative charge. It gives them the power to strip the electron from that hydrogen. So they're really good at making an acid. And you're gonna see later on how using this hydrogen is gonna help us create reactions that can move things together. So big 3 players. Let's go. So when we're talking about DNA, this is where these groups are gonna come in. The main one I want you guys to focus on is gonna be on our amino groups and our phosphate groups. So amino groups, these blue ones, you see it's a nitrogen with the hydrogens. Those, again, big difference in electronegativity. Nitrogen is gonna be greedy, takes the electrons, aka the hydrogen is gonna have that positive charge. You can see it right here. Because of that negativity, you can actually either pick up hydrogens or kind of hold on to them, and that'll be like a little bit of creating a little bit like a positive charge. We use that one a lot in DNA. The other one we use is going to be this guy, which would be our phosphate group. So for this one, you're going to see a P, and it's basically engulfed by oxygen groups. P is a little funky because it likes to have it binds to 5 different partners. So you see 1 bond, 2 bonds, 3 bonds, and there's your double bond to get to 5. This thing, like have you guys ever played with magnets? You know, like if you hit the positive end, negative end, they'll kind of like have a force where they pull on each other. But then if you put them backwards, you go negative against negative, you can push and push, and it'll be hard for you to get them to close. When you have a phosphate with these oxygens like that, that's the kind of thing I want you to be thinking about. The thing is so negative that if I put another one right next to it, those two are kind of going to be trying to push it away from each other. So I'll show you how this is going to be really critical for energy. All right. And then the final group, the boring group, it's the methyl groups. Remember, this one is carbon hydrogens. This one.
Can you go back to the phosphorus?
This one.
Why does it have like a -2 just like in the name for phosphorus?
Perfect. So for this one, we're thinking about kind of you're balancing your protons and your electrons, right? And what you're going to see is this oxygen is being fair in the sense that it's sharing the 2 that it needs, and it's going to be okay, right? This one, when you're looking at this oxygen, you see it only has 1 bond, but it will have an extra electron surrounding it. So
that oxygen is just very negative because it has an extra electron.
Normally, you'd expect to find it kind of with an H group there. And the H are kind of canceling it out in the sense of it almost feels like you're talking here, like almost like an ion in a cation, like with salt that are in water. They strip the electron, and now this thing's going to have a charge on it. So, because you have 2 oxygens that don't have their hydrogen groups, they have that extra electron. That's what's giving us this -2 charge. And then, but when we see it forward, kind of keep an eye on it, you'll kind of see that if they bring in hydrogens to this, they'll make this charge start going down. So if they had put like OP, O3H, it'd be a -1. Okay, HH, you bring it in. Yeah, good question. Your name starts with a J, right? No?
Yeah.
Yeah, what's your name?
Jacob.
Jacob, all right, perfect. I forgot to study this morning, guys, so I'm not too fresh on your names right now, but next time I'm gonna sure be on it. Any other questions? Oh, pretty good. All right. So again, methyl group, this is the one, it's the boring one. It's the one that doesn't want to play with water. And here we're kind of showing you this is a little ring. This is part of of your DNA base pairs. This is the main chain that we're kind of following, this carbon thing. And you see we've attached this thing on the side of it. When you attach this, the only thing you're doing is you're bringing in something that's non-polar. It doesn't want to interact with things. So sometimes you'll see that in our bodies, we use these to turn things on and off based on whether our body's normally wanting to interact with a polar thing, and then you put this, it won't let it do it anymore. So these are pretty cool, super boring. I normally don't draw them. Everyone, when we're doing it, we're kind of lazy, we leave them out. So whenever you see my drawings, you're gonna see that I'm just gonna do little sticks, and it represents basically carbon with a bunch of hydrogen, or with their hydrogen set up. All right, so we talked about the power of hydrogen. Now we're gonna get into just quickly, we're gonna go over some of the bigger molecules that we can make out of it that matter to us, right? So these bigger molecules are just a mix of things. Here you can ignore, this is just a crazy picture of a protein. But let's zoom in, let's talk about first what really matters, right? Which is energy and sugar. So right now we're gonna talk about kind of the different ways you can find sugar. So what we can do is we can find these bigger molecules. We'll see that they kind of have repeating units, like they'll have almost similar shapes. When they're by themselves, we'll call them monomers. And then as we glue these molecules together, we'll have like a polymerization event, then we start making the polymer, right? But it will be repeating units of the same thing glued over and over next to each other. So for this, here you can see this is our sugar. Here's the fancy way to draw it. You can see there would be your first carbon. That's why you get to number 1. In order to complete its 4 bonds, there you can see an H group. There's our hydroxyl, right? Then we go to carbon 2, carbon 3, carbon 4. And again, you're still seeing the same thing, right? Hydroxyl group, H group. This will be number 5, the 5th carbon, and your 6th one kind of hangs up here. So this will be the individual unit. So that's our monomer. And then down here, we're looking at how we can actually glue them using carbon 1, carbon 4, these hydroxyls. We can use that energy to kind of help us link up into the next molecule, right? And now we've basically linked these up with actual covalent bonds. And now this is the new molecule that we care about. So for this one, these 2 words, dehydration reaction and hydrolysis, put them bold, put them in your dictionary in the back. They're going to come up more than several times on how we glue things together. And you're going to see that we kind of use the same trick over and over. So again, let's go back. We're looking at 2 different sugars. These are monomers, right? So this is one sugar with 6 carbon. Here's your other sugar. There, carbon 4. There's carbon 1. We're going to try gluing these 2 together. What's going to happen there is basically this OH is going to break its bond with this carbon. It's going to pull over the hydrogen to itself. At that point, you're going to have an O with 2 H's. This other O, which gets left behind, is the one that now is going to be missing a partner, and the carbon's missing a partner. So they jump in and connect with the carbon. Right? So this is called the dehydration reaction, because the big thing is that we're pulling out water from the system to make it glue itself. Right? In order to kind of fight that, this will be the opposite reaction, right? Which will be hydrolysis. So it'll be now we can use the water to break these things apart from each other. So here we have our dimer, or disaccharide, which is 2 sugars that are covalently bonded together. And what we can do is we're going to bring in the water, and one of the H's is going to get shifted to this O that was already there. The other OH group is going to attach to the carbon. And now we've broken this back to sugar. So in our bodies, we're doing these 2 reactions all the time. Like when you have too much food or too much sugar in your system, this is the sugar that our body knows how to break and get energy out of when it's a single sugar molecule. If we have too much and we need to put it away, our cells will send out insulin, and they'll tell them, hey, glue these things together to make these longer chains. This stuff, we can't digest it. Like our body can't instantly take the energy out. So this will be a way that we can store our energy. A few hours later, you have an 8, your sugar levels start to drop, and then your body can decide to go undergo this process, right? You have these longer chain sugars, you add water, you break them up, and now this is the things that our body can actually digest. So yeah, so quick story, right? That's how we basically, when you're looking at the foods, the things that we eat, in plants, the big thing is they have cell walls, and then the cell walls, sorry guys, I'm gonna go back to it, sorry. They basically, the plants, their job is they use sunlight to glue carbon together to make sugar rings. The sugar rings, and they have different functions for them, but that's really what they're really enriched in. So whenever you're eating like a fruit or veggie, bread or anything, you're really just taking in a bunch of sugar. So this slide is a little complicated. We'll walk through it kind of fast. The only ones that we're talking about here are going to be different types of sugars. They have these fancy names where it's like, hey, 3 carbons, they'll call it a triose, 5 carbons, or 6 carbons. To be honest, the one that I'm gonna bring up a lot is gonna be this guy, a ribose, and that's because I love talking about a ribose when it comes to our DNA, right? Because like ribonucleic acid is gonna be a sugar inside that guy. The other one I talk a lot about is gonna be glucose, which would be my 6 carbon ring, For this one, 6 carbon ring. This is really important because this is how we power up our stuff. You don't have to memorize these hexos, pentos, and trios. You know those for now. Pretty good, yeah. So this is one way to draw them. I normally don't draw them like this. If I'm ever drawing these, you're gonna see I'm gonna get pretty lazy, or I'm trying to be fast. So whenever you see these, you're gonna see me, for example, for these, I'm gonna draw something like this. And I'm already, like in my head, I'm imagining, you're seeing all this stuff, it's already here. Right? You have your H's, your OH groups, H, OH, but I'm only gonna do that ring. For the 5 one, I'm basically gonna draw something like this. It's gonna be 1, 2, 3, 4, and it's a little stick hanging. Yeah? So, if you ever wanna go back to look at them, you can go to here, but the cool thing is, again, look at those functional groups showing up, and those are gonna be able to give us different little tasks. So for example, here we have a 6 carbon chain. We start from there, 1 through 6. We have that double bonded O at the top, which is our carbonyl group. That has really strongly localized a negative charge. You have that double bonded oxygen, no hydrogen. You can basically use that one to steal this hydrogen to itself. And instead of having a double bond, now it switches over to the single bond. You have an OH group. But since this is free, it'll actually try to make a connection with this carbon because they'll lost the connection. The whole point is that when they start out, they're stable, right? Carbon has 4 partners, oxygen has 2. And when you're done, that carbon is going to replace these. See, it has 4 partners, and oxygen has 1 and 2. So you can basically transition to, from a stable state, you have this little swap of elements, and you can go back to the stable state. Fancy version, I rarely draw that. You're going to see this a lot from me. So that's going to be pretty cool. All right. So this one, I could almost skip it. These are very specialized words, but it's the same idea of monomers and polymers. That's the thing I want you guys to kind of stick with, right? Here, the fancy word is monosaccharide means single sugar. Disaccharide means 2 sugars, right? You can have a polysaccharide, which means just a bunch of sugar. I'll pick less on this little keywords. I'd rather you guys be really comfortable with monomer and polymer, right? But we'll see, again, that same reaction. Here's our OH groups, our hydroxy groups, we're gonna have them react. Let's say, we'll pick this one for now. This OH group is gonna steal back hydrogen. That's what's gonna give us this water. This O is the one that's gonna stay behind. And that's the one that's gonna, when it's trying to find some partner, it'll link up these molecules. So the same thing. In this case, we're moving to the right. This bond that we made is called a glycosidic linkage, right? Should have done this earlier, sorry guys. So what's the name of the reaction that just happened right now? Write it on the little card, and then put your name on it. This will be our little quick little clip. What do you guys call that reaction? Going from basically going to right.
I don't know what you're asking for.
So we're going from a monosaccharide, and we're doing chemical reactions that's going to make it into this double sugar. Right now, before we were talking about the 2 different styles of reactions we can have. What are they called? Or which one are we doing for this? So we'll make this really easy for me to grade. So we'll just talk about it real quick. So we're going to appreciate right now, right? There's our bonds, and what's coming out from this is going to be the water. Right? So when we're talking about the reactants, normally they talk about this product, right? Either water's going in or water's going out. And that's why we use those 2 key words for this reaction. if you're talking about the bond, then you focus on the green thing. The glycosidic linkage, I've seen them throw it in a few tests because people like it. So just be comfortable, which just means you're keeping track of the oxygen that got glued to keep the sugar together. But honor code, right? Look at your paper. What did you guys put for this one?
Dehydration.
Right, dehydration, perfect. You're taking water out. Awesome. If I had invested you guys, we would have been the opposite. Wanted to break these things.
Hydrolysis.
That was perfect. All right, cool. So polysaccharides, again, I will not make you memorize these kind of fancy words, just polymer, monomer, or dimer, those are the cool ones. This is super special sugar, but that's the whole thing that, a real powerful thing behind it is like, this is the place where I really love the way that it works. If you zoom in and look at an individual cell, it builds a cell wall, it's the coil cell. And what that cell wall is basically these like super long cables that are just sugar molecules glued to each other. So they're really strong chains. Even though it's just one small covalent bond here, the thing that makes that fiber really strong is that we have OH groups like this hanging out, right? OH groups, aka we have 4 interactions. This thing will then clamp into itself with those hydrogen bonds. And just because you have so many of them in a row, this thing's gonna stick to itself. This is like the craziest version of Velcro. This thing is clamped off to itself. It's super powerful. Like one of the things we were doing, we were trying to figure out how much pressure this tissue had, like when we were looking at the plants. And we were trying to like cave in cells. And then the pressure we got to was about 40 PSI. Like that's like your heart pressure. And that's how much force that bubble is. The cells inflated on the inside and it's wrapped with this sugar. And that makes it super tough, but we can't put into it unless we get above 40 PSI. But that's the toughness. Our plants actually work on themselves. Like if you see the plants and the wind hits them and sways them, they'll purposely try to make more of the cell wall material to strengthen up the cells in that region, and that's how they stiffen up. And what we do when we eat them, we basically start breaking up these sugars. Sometimes they're linked up in different ways, and we have trouble digesting them, but for the most part, we just start shredding these back to the 6 carbon rings. And we're good to go. All right. The other macromolecule we're going to talk a little bit about is going to be about lipids or fats. So for this one, you're going to see that it's amphiphatic. So the ones that we're going to talk about. Oh, sorry, I pressed it. So first, let's look at this lipids. So we're going to look at triglycerides. Triglycerides. In this case, right, we're going to go through the drawing pretty fast, which is down here, you see the super simplified version, just the sticks. It means there's a carbon, 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 all the way up. And what you guys are looking at there, what you can imagine would be carbon with a bunch of hydrogens, aka non-polar. This thing does not like to play with water. But you can see up here, we have that functional group, double bonded O. There's our OH group. So we have our carboxyl group. That has those negative charges reactive. And look, we're doing the exact same trick. There's your OH, there's an OH. This is going to steal that H, right? And that's going to be the oxygen that's left behind and try to bond to this carbon. So again, another dehydration reaction, right? Pull water out of the system, it can do itself. You can imagine in our bodies, we can do the opposite as well, which is if we add water to this already bounded structure, we can bring it back and break it into something. So this is a really cool one. So here we're looking at, I don't know, you guys probably heard this in your food or you're reading about things of saturated and unsaturated fats. So the cool thing about this is we're still here looking at focus on the behavior of carbon itself. But in this case, we have carbon, and we see saturated, in my head I almost think it's saturated with hydrogen. That means that every carbon is full of hydrogens, right? So this one has 3, that one have 2 hydrogens all the way across, meaning that you have a single bond every single place that you go. The cool thing about like the single bonds for carbon is in 3D space, it does this weird thing where it's like, if carbon's here in the middle, it's able to bind to something, say 1 bond to a hydrogen there. And it makes it a little bit like a tetrahedron like this. Okay. So these are kind of be spread out like that. And you have another one like this. These would be your hydrogens. That's how we used to draw in chemistry, hydrogen. And hydrogen. So this one is perfectly vertically up. This one's kind of sticking this way. This one's kind of sticking this way. And this back one, imagine it's sticking backwards. So it opens them up. Like imagine my core was a carbon. My legs would be the hydrogen. It'll set one up like this, and it'll set up the other one like that. That means that because you have one bond and you have the freedom, these things are super comfortable in rotating. So when they're getting made, they're going to try to find this super stable conformation of doing this. Where they'll be like, hey, let's get the carbons and the hydrogens will be away from each other. Everything's happy. We literally bring in a single double bond right there. That double bond, instead of being in this 3D thing of this is up, these two are forward, that's backwards. It puts them all on just a single plane, and it looks like this. So in this case, these will all be like almost laying flat. This will be exactly 180 degrees from each other. So now instead of having like the super fancy, this kind of setup, it's basically double bond and it goes like a Y. When it does that, this double bond won't let it twist. Because it doesn't let it twist and you stick it into 180 degrees, you're gonna cause this weird bend basically on this side. This should have been coming back down. Instead we have this weird angle. This will make this thing bend into a weird shape. And literally that kink's gonna mess with this guy.
So are saturated always pyramidal and unsaturated always planar in structure? Like
Yes, right, because when you're saturated with hydrogen, 4 hydrogens, that means you can do those 4 connection spots. It's a hindered, right? When you have unsaturated, it means you've lost some hydrogen and you're using this double bond. Now it's going to be this planar 180. So it hurts us in 2 ways. This angle is going to get limited, and the other one is when you have a double bond, you can't spin around. So it'll kind of stick you one way and you're going to pop down. And really, when we talk about these two, what ends up happening is, right, when everything's hot, it'll be bouncing around. So it'll be in this liquid. As things starts to cool off, they'll try to settle, these molecules will settle into themselves, and then they'll basically form a crystal structure. When they're saturated, it's very easy for these to kind of clamp onto each other. They'll have a little bit of like a van der Waal interaction, that these molecules can stick to each other, they collapse. That's why like that stuff is solid at room temperature. Right? At the exact same temperature on this side, this little kink doesn't let them stack into each other properly, so it's hard to build a crystal form, and that's why we can't get them to actually turn into solid form at room temperature. If I cool them off enough, eventually I can make them collapse, but for the most part, you're gonna pick them up something like this. So even though it's just a double bond, we haven't messed with polarity or anything, we can really affect the way that it functions. Yeah. All right. That one should be good. All right, and this one you guys should be all familiar with, right? This is the one we did on Wednesday. So we're looking at another fat. This is going to be a little fancier. Again, the bottom hydrocarbon part, it's a bunch of carbon, a bunch of hydrogen, non-polar. In my head, I'm always thinking that's the boring part. Right? It won't, this one's made with water. And then you have the top part, where again, there would be the functional groups we care about, right? P with a bunch of O's around it. And on top, you see we have our nitrogen with a bunch of carbons. They all have polar interactions, aka they'll make their own little spots of being charged positive or negative charged. And that's going to give us the ability to make these kind of structures, where when they organize themselves, the gray parts, which are hydrophobic, will hang out with each other, but they don't want to hang out with the water. This little blue caps on top, you basically need these groups up there. If you love to play with water, they'll be able to interact with it on that side. And instead of this molecule, if you just grab one of these, stored in water, it'll be super unstable, because these things wouldn't be happy interacting with water. They'd be getting tossed around. The moment they get into something like this, everything's happy, right? These little legs are hanging out with more hydrophobic stuff, so they're happy with each other. Even the bottom of them, they're kind of clamped into more hydrophobic stuff. And then the water level stuff is basically at the edges. So super powerful stuff. What is this one? So this is Jacob's question, right? So check it out. There you have a phosphate group, right? In the other one, it didn't have that extra bounding partner up here, right? So we had O minus and then another O minus. Now that we've had the bonding created, now we just put a charge of -1. So you can see we can kind of mess with it. You stick a hydrogen there, we can basically cancel it out. All right, cool. You guys can watch that one afterwards. And the other one we're going to talk a little bit about is going to be the steroids. For this, that big thing we're going to look at is going to be basically they have these like carbon rings, and that's going to make them a little bit shorter than the other ones. But these functional groups are basically going to give it a little bit of like water loving ability. So when we look at steroids inside a plasma membrane, they'll have the polar functional groups kind of hang out here, and their body hangs out a little bit in the inside. It might be like a third or a fifth of the height of one of these, so it kind of messes with the shape of it. Like all the legs on the clamp up, and this thing's just top heavy. So I'm using some pretty cool stuff for it. All right. Then one of the last molecules we're going to talk about is going to be basically our DNA. I'm going to go over this one more than a few times. So for this, let's go over the different parts, right? So on this side, when we talk about energy, we're going to have those phosphate groups, and you're going to see we're going to have those of super high enrichment of oxygens. Right? There, I want you in your head to be thinking again, those magnets with negative impulses, they're pushing on each other. So these three things is you build up a molecule that has a bunch of negative things clamped up in a really tight space. So if you find anything positive, it's gonna want to jump off, and when it jumps off, it actually transfers energy. So when you guys hear ATP, the big emphasis there is triphosphate. Those are our energy stuff, right? Next part we're gonna pick up here is gonna be, here's our sugar ring, right? So in this case, 1, 2, 3, 4, 5 sugars. So this is what makes our ribose. If we change one of these oxygen groups with either DNA or RNA, in that case, that thing gets repeated over and over. And then this last thing is going to be our nitrogenous base, right? So in this case, we see there's 2 little carbon rings. There's one here, one here, and we have different nitrogen groups. So this is where on our DNA, we'll have 4 different base pairs, and each one has its own little arrangement of how these work. A and T, they inertly balance where one has 1 circle, the other one has 2 circles, and you can bond to each other. C and G are the same thing, and they only partner up with each other. So CG is a set, AT is a set. And based on the little differences of those functional groups, our body can literally detect the different base pairs and write different amino acid codes. You can write different RNAs, all based on those functional groups. The last molecule that I care about is going to be proteins. I'm gonna have it here for now, but I'm gonna move it once we're done with the class, which is, we normally, in our cells, they have a complete copy of our whole DNA, right? That's the instructions to make every single amino acid that we could ever want. Our body then turns only a snippet of that code into RNA, and that RNA code gets made into amino acids. Those are the ones that make those 3D like molecular machines that actually do things. And the ways that those get specialized is they have this thing called a functional group, or like an R chain, where here we can see basically the different versions of those. So in the backbone, we have nitrogen, a carbon, and then a carboxyl. These will actually do that same reaction of putting out water, and we can glue these together. And then on this side, depending on what amino acid I pick, I can get different things, right? Like this one only has a carbon with hydrogens, aka it's non-polar. It's not going to want to stick to water. But then I can switch it out with some fancy thing like this guy, right? So you see the chain has 1 carbon, 2 carbons, and now I throw in a carboxyl. So now we know this thing's going to be able to act like an acid. You can jump to hydroxyl group. There's your NH3. Depending on what we add, it'll give us the power to do different things. Grab onto positive things, grab onto negative things, or hang out with non-polar stuff. Don't memorize it. You never need to memorize this thing. I just want you to know that there's this group, this bond, and that's it. Yeah. So I'm gonna kick it off to the side. That way you guys don't go too crazy with it when you guys are studying. And then talking about that, so I know bio has a lot of vocab and it takes a lot of time, right? So before I used to try to read the book when I first took the class, and I would try to memorize every single line, every single word. every single thing. And at that point, I realized my brain can't handle that much information. Like, I'll just read so much, and my brain's eventually like, it stops paying attention. Until I had this one TA who helped me, like, basically go through the notes, and he would delete slides to tell me, hey, this is really important versus this thing is not that important. Like today, we talked about this monosaccharide, disaccharide stuff, and I was making it clear to you guys, I'm like, you don't have to memorize it. I mean, we do our study, and then he's like, hey, get rid of these slides from your study deck, right? The things that I emphasize, which is the monomer, dimer, the name of the reactions, right? The hydration or hydrolysis, those are the ones that like make a new set of your PowerPoints, and you're just keeping the important ones. That way you guys can initially read the book, kind of like if you're reading just a regular story, read through it, get a gist, come to class, see how much you get, wherever you don't feel comfortable, that's stuff you go back and put emphasis on it. Or if you see that I emphasize it a lot, make sure you guys try to do the same. For the review session, what I'm going to try to do is I'm going to grab all the slides that we have for the class, throw them into one big PowerPoint, and I'm literally going to sit there and delete a bunch of slides. So if something I think I'm like, hey, there's 4 slides that explain how this one thing behaves, but what I care about is the behavior, I'm just going to keep this slide, right? You then go study the slide. It's like, if it makes sense to you, great. If it doesn't, go back to the original PowerPoint, and you can go look at the slides before it to kind of get info of how do we set up this stuff. That'd be my biggest hint for you guys for studying. And then the other one, of course, is study with each other. The tutoring service, they emailed me yesterday. I think they have like 12, no, 8, maybe 8 different tutors available. A lot of the kids were tutoring, they've taken the class with me before, so they literally know exactly how I do quizzes, how I ask questions. They'll be able to help you guys and give you tips on how to study for me. They can go over how to set up quizzes the way I ask you for things. And so reach out to them, of course. Also, you can stop by office hours and talk to me. The other thing I was going to bring up was my exam format. So when I do the exams, normally I break them up into 3 sections. The first section is going to be vocab. It'll literally be like a little statement or a little question with a word bank, and you're matching it up, right? The second part is going to be free response. It might be like 6 to 8 questions, but they're short. Like my goal is you answer it in 2 questions. If you forgot the keyword or you forgot what it was about, and you have to give me more definitions, then make it a little bit longer, but don't try to go too crazy with them. The last part, part C, is going to be the long response. That one might be 1 or 2 questions, and at that point, I'm expecting like half a page answer to a page. Yeah. So you guys have any questions for today? No? All right, cool. So let's call it, and then I'll see you guys on Monday.